basically, there's quite a lot of methods. And as we've done before, now we've done the quality control, dimensionality reduction, integration, and clustering. And we want to get to the differential expression. In Seurat, we have a function called find all markers. In Scran, we have find markers. And in ScanPy, we have ranked genes groups. And basically, there's actually also a function called find markers if you just want to compare two clusters. But find all markers will do this one versus all for whatever you have in your active identity slot in the Seurat object. So after you're done clustering, the last step of clustering you did will always be your active identity. And the slot should be data. And the assay, if you run CCA beforehand, make sure that assay here is RNA. You don't want to do differential expression in CCA space. You want to do differential expression on the log transformed count, log normalized values that are in the slot data. You don't want to do it on the scale data object either. And then there's, yeah.
Okay, so for clarification, so always the raw count data?
Not the raw count data.
Okay.
There are some specific tests that require the raw count data, but in most cases, it is actually the log normalized data you should run these tests on, especially if you do this rank based or something.
Okay.
Well, for the rank base, it shouldn't matter that much, but the log transformed would be the standard to use. I'm not sure if any of the Seurat methods actually changes. I know there are some that says it should be counts, but I don't think any of those in Seurat are those.
Yeah, by the default will be the data normalized one. Only the stack, negative binomial, and Poisson, and maybe must will use the
must you can do. You should do on the log transforms.
Yeah. Okay.
And then you can
What if they're SCT normalized?
You still do the differential expression. I would still do on the non-SCT data, I think.
Okay, thanks.
And then there's a lot of cutoffs. We will only, and this is mainly for speed up. So they first calculate the log fold change for all genes and discard anything that's below 0.25, that we don't even test those. And where at least 10% of the cells in at least one cluster express the gene, we can, the minimum number of cells that has the gene and so on. So there's a lot of cutoffs you can define. If you want all genes to be tested, it will be slower, but you can just set all of these to 0, all these cutoffs, and also the return threshold to 1 to report all genes. And also this only positive by default is false, so you get both up and down regulated, but it's much faster if you run it with true, because then you have fewer genes to test, if that's what you're only interested in. So, as I said, Seurat has a lot of different methods to run for differential expression. The default is Wilcoxon rank sum test. And I'm not going to go through all of these, but we'll talk a little bit about statistical inference. And the idea is that really decide is what we observe, the differences we observe, just due to random noise, or is it a true signal, more or less? So we can model our data in a lot of different ways and assume different things about the error we can have. And of course, the more data we have, the larger population we have, then the conclusions we can make are better. And this is also something we talked about before. How many cells do we need to have? And if we have too few cells, we can't say a lot of confident things about differentially expressed genes from 5 versus 5 cells. So, as an example, is there a difference between height for the students from last year and you guys? So the null hypothesis would be there is no difference, and then we can have an alternative hypothesis that there is a change. And then we can look at the values. This is probably not the true values. And we can decide what kind of tests we want to do. In this case, we have really nicely normally distributed values, so we can use a t-test and calculate the significance of the overlap of these two distributions. So the better the model fits your data, the more accurate the statistics is. But we can also use non-parametric methods that doesn't assume anything about the distribution of our data, which the most commonly used is this Wilcoxon or Kruskal-Wallis or Kolmogorov-Smirnov, that's hard to say. So mainly we would just convert the expression values to ranks. So ordering the genes by their level of expression, and then test where between two sets of cells or groups of cells if the ranks are different, clearly different, so that this gene has a higher rank in this group and a lower rank in another group. So it's sort of the relative expression of different genes. The issue can be a lot of ties when we have a lot of zeros. And if we can have a parametric test that fits our data well, it probably will be more powerful. So that's the Wilcoxon and the t-test we talked a bit about.